All right, guys, so this is chapter 16. I'm just going to cover the lymphatic and the immune system. So they really work hand in hand to protect the body. The immune system consists of cells that defend the body against disease, and most of these cells exist within the lymphatic system. So that's why we're going to talk about these two systems together and why they're really in the same chapter. So here's a visual of the lymphatic system. Consists of lymphatic vessels, lymph, which is actually the fluid within the vessels, lymphatic tissue, and lymphatic organs. So the vessels of the lymphatic system cover the body in much the same way as blood vessels, which we just talked about in the last chapter. But unlike blood vessels, lymphatic vessels carry fluid in only one direction, and that's really away from the tissues. So it carries it from the tissues back to the heart, okay? The tissues and the organs of the lymphatic system include the lymph nodes, the thymus, tonsils, spleen, and red bone marrow. So what are the functions of the lymphatic system? It plays a big role in fluid balance. Fluid continuously seeps out of capillaries into surrounding tissues. Capillaries reabsorb about 85% of the fluid, leaving about 15% behind. Over the course of the day, that would total as much as 4 liters of fluid, and would certainly cause some havoc on our body. So one of the roles of the lymphatic system is to absorb this fluid and return it to the bloodstream. It also plays a role in absorption of fats. So there's specialized lymphatic vessels in the small intestines that absorb fats and fat-soluble vitamins. Probably the least one we're going to talk about. And then it also plays a role in immunity. So lymph nodes and other lymphatic organs filter lymph, which is, again, the fluid inside the lymphatic vessels that it picks up from those capillary beds, and it removes microorganisms and foreign particles. So let's look at what these lymphatic vessels look like. Okay, so we're going to look, somewhat similar to the veins in that they have these nice thin walls and they have valves to prevent backflow. But here they're filled with lymphatic fluid or lymph. Okay, it's clear, colorless fluid similar to plasma but with a lower protein count. And again, it's the fluid left behind after capillary exchange. Unlike the cells in the veins, which are tightly joined, the cells that form lymphatic vessels' walls overlap loosely, allowing gaps to exist between the cells. Fluid can thereby enter the lymphatic vessels between these overlapping epithelial cells, so it serves a purpose. And then again, we have these valves to help prevent backflow and ensure that the lymph kind of moves steadily away from the tissues and towards the heart, because again, they're kind of working against gravity. Here you can see a little bit more about the flow of lymph. So we get lymphatic vessels that start here in these tissue spaces. So this is the capillary bed, okay? So this is the end of one of the lymphatic vessels, okay? So the tissue fluid flows into the vessels through the gaps between the cells. We also get things like bacteria, lymphocytes, and other cells flow in this fluid. So they get into the lymphatic system. The vessels converge to form larger and larger vessels. Periodically, the vessels empty into lymph nodes where immune cells actually can phagocyze bacteria, which is their job. The vessels continue to merge, eventually forming lymphatic trunks, which drain major regions of the body. Okay, so the lymphatic trunks converge to form 2 collecting ducts. We get one near the right subclavian vein and one on the left. Okay, then this is where the lymph joins the bloodstream. So it gets back into the blood flow, okay, and moves back to the heart to be, again, transferred throughout the system. Okay, so it kind of rejoins the blood flow. And again, the skeletal muscle pump and the respiratory pump both help to increase lymph returning to the heart, just like we talked about in the last chapter. So let's watch our first animation here.
The lymphatic system consists of lymphatic vessels, lymph, lymphatic tissue, and lymphatic organs. The vessels of the lymphatic system cover the body in much the same way as blood vessels. Lymphatic vessels are filled with lymph, a clear, colorless fluid similar to plasma, but with a lower protein content. Lymphatic vessels have thin walls and valves to prevent backflow. Vessel walls are formed by a thin layer of epithelial cells. Unlike the cells in veins, which are tightly joined, the cells that form lymphatic vessel walls overlap loosely, allowing gaps to exist between the cells. Lymphatic vessels originate in tissue spaces as microscopic, blind-ended sacs within a bed of blood capillaries. Tissue fluid, which is the fluid left behind after capillary exchange, flows into the vessels through gaps between the cells. Bacteria, lymphocytes, and other cells flow in with the fluid. As the vessels progress on their path toward the heart, they converge to form larger and larger vessels. Periodically, the vessels empty into lymph nodes, where immune cells phagocytize bacteria. The vessels continue to merge, eventually forming still larger lymphatic trunks, which drain major regions of the body. The lymphatic trunks converge to form two collecting ducts, one near the right subclavian vein and one near the left subclavian vein. Lymph joins the bloodstream when the collecting ducts merge into the subclavian veins.
So you can see on this diagram here actually that, again, the lymphatic system kind of functions in two areas. We get a systemic and we get a pulmonary. So the pulmonary is going to drain over here, and the rest of the system is going to drain over on this side. And that's what this is going to kind of show you here, okay, that we get the right lymphatic duct drains lymph for the upper right quadrant of the body into the right subclavian vein, and the thoracic duct drains lymph from the rest of the body into the left subclavian vein. So the lymphatic organs, again, are red bone marrow, thymus, lymph nodes, tonsils, and spleen. Okay, the primary lymphatic organs are the red bone marrow and the thymus. They provide a location for B and T lymphocytes to mature. Then the lymph nodes, tonsils, and spleen are secondary organs. They contain lymphocytes that have already matured in the red bone marrow. We'll talk about these in a little bit more detail here. So we already learned about the thymus before, an endocrine, but it's located in the mediastinum. It's quite large in children, but begins to shrink by age 14. Or at age 14, excuse me, and then by adulthood, it is quite small compared to its original size. It's divided into lobules that extend inward from a fibrous outer capsule. Each lobe has an outer cortex and a medulla, and these are filled with those T lymphocytes. As I said before, immature T lymphocytes travel from the red bone marrow to this thymus, okay? Inside the thymus, the cells are protected from antigens in the blood, giving them a chance to divide and mature. So we want them to get to the thymus so they can mature enough to do their job. So as they mature, they kind of head inward into the inner medulla. And as they encounter different lymphoid cells here, this is where they kind of get training in how to distinguish cells of our bodies from foreign cells. And so then once they're kind of trained in the thymus, they're released out into the bloodstream.
The thymus is a primary lymphatic organ that provides a location for B and T lymphocytes to mature. The thymus, which is located in the mediastinum, is divided into lobules that extend inward from a fibrous outer capsule. Each lobule consists of a dense outer cortex and a less dense medulla filled with T lymphocytes. Immature T lymphocytes, also called T cells, travel from red bone marrow to the outer cortex of the thymus. Inside the thymus, the cells are protected from antigens in the blood, giving them a chance to divide and mature. The developing T lymphocytes migrate toward the inner medulla. As they do, they encounter other lymphoid cells, such as macrophages and dendritic cells. This process trains the new lymphocytes to distinguish between the cells of its host body and foreign cells. Once the training is complete, the lymphocytes are released into the bloodstream.
So the thymus is kind of the training center for immature lymphocytes. The lymph nodes, again, are another portion of the lymphatic system. We have hundreds of lymph nodes throughout our body. Some are really small, like 1 millimeter, and others are up to an inch. So what happens in the lymph node is lymph passes through them. It kind of slows to a trickle, and it slows so that the lymph node can do its job of removing pathogens and other foreign material. It also serves as a site for other types of lymphocytes. You don't need to know all of these different pieces here. Basically, what you do need to know is that it cleanses the lymph fluid. So we kind of mentioned before that it might bring bacteria here to be engulfed and killed, and it really cleanses the lymph fluid. So that's what lymph nodes do. You might have heard about lymph nodes before when talking about cancer, and what happens usually is cancer cells break off from their site. They end up in the lymph system, and they get to the very first lymph node closest to the cancerous site, and they start to divide here. So that's why we check lymph nodes often for cancer, and that helps us to determine if the cancer has spread. Tonsils are another section, so we actually, these are really just masses of lymphoid tissue. There's 3 different tonsils in the back of the throat. They all do a job against guarding against sorts of foreign materials or bacteria or viruses that enter through our nose and throat. So we have a single pharyngeal tonsil, also known as the adenoid, sits on the wall of the pharynx just behind the nasal cavity. A pair of palatine tonsils, which are what most people are familiar with, lies on the posterior side of the oral cavity. So we get kind of one on each side there. And then we have lingual tonsils in patches on either side of the base of the tongue. The palatine tonsils are the largest, most prone to get infection, which leads to tonsillitis, which is why people ultimately end up having them taken out. The spleen, which is about the size of a fist, is our largest lymphatic organ. Just like lymph nodes, the spleen has this kind of fibrous capsule, and it has these kind of inward extensions as the spleen divides into different compartments. Okay, 2 types of tissue here. We have red pulp and white pulp. See this telling us here. White pulp contains lymphocytes, okay, and the red pulp contains red blood cells, or sinuses filled with red blood cells, basically. Blood collects here, and then it returns back to the heart. So lots and lots of blood flow to the spleen. The spleen is actually really easily damaged, and so people have to take care when there's a traumatic event that there wasn't splenic damage, because people can be hemorrhaging internally. People can live without their spleen, but certainly they're going to have a decreased immune system, because that's really one of the major jobs of the spleen is immunity. Okay, lymphocytes and macrophages live here in the white pulp. They screen the passing blood for foreign antigens, so it's kind of like one step in the immune system, and it helps to destroy microorganisms. Also, the spleen plays a role in destruction of old RBCs. They can remove and digest worn red blood cells and imperfect platelets. It also stores 20 to 30% of the blood's platelets. Okay, so again, that's why we have such an issue with bleeding if it's damaged. And also the spleen produces red blood cells in the fetus. So that's one of its jobs in the fetus. So we talked about the lymph system, pretty straightforward. Just need to memorize the different parts of it, where it flows to, but hopefully we kind of already get that a little bit. The bigger section here, or the more in-depth section, is the immune system. So there's going to be lots of pieces to the immune system that we really need to spend some time learning. So there's 3 lines of defense. We have the first, which is an external barrier. That's our skin and mucous membrane. So they keep most pathogens outside of our body. Second line of defense, so if the pathogen does makes its way through the first line of defense, the body launches what we call non-specific immunity. It involves mechanisms aimed at a wide variety of threats, so it's non-specific to the type of bacteria, virus, whatever that's coming into the body. The third line of defense is specific immunity. So this is when the body has a memory of the pathogen because it's fought it before, and we get exposed to it again, we know how to recognize it and can target a very specific response. So non-specific immunity is a little bit more generalized. So let's talk about how non-specific immunity works. multiple different ways. We have phagocytosis, antimicrobial proteins, natural killer cells, inflammation, and fever. So they're all aimed at a broad range of attackers or threats to our system. First one is phagocytosis, okay? So what they do is they actually ingest and destroy microorganisms and other small particles. When a phagocyte encounters a microorganism, it sends out membrane projections called pseudopods, or these kind of false feet. They envelope the organism, forming a complete sac around it. It travels to the interior of the cell, fuses with a lysosome, which contains digestive enzymes. The digestive enzymes destroy the microorganism, and the phagocyte actually can expel the waste products. So this is pretty interesting. I think our immune system is very interesting because we have so many ways which our body fights off organisms.
Phagocytes are cells that ingest and destroy microorganisms and other small particles. If a pathogen makes its way past the skin or mucous membranes and enters the body, it will immediately be confronted by phagocytes. When a phagocyte encounters a microorganism, it sends out membrane projections called pseudopods, or false feet. The pseudopods envelop the organism, forming a complete sac called a phagosome. The phagosome travels to the interior of the cell and fuses with a lysosome. The lysosome contains digestive enzymes that destroy the microorganism.
So pretty neat. The other piece of this that we have different types of phagocytes. So we have the most important ones are neutrophils and macrophages. Okay, neutrophils roam the body versus most macrophages remain fixed within kind of strategic areas where we have a lot of potential for microorganisms to come in. Neutrophils are summoned to an infection by a chemical released from inflamed cells, which is chemotaxis. So let's watch the animation on this and see how that works.
In a process called chemotaxis, important phagocytes called neutrophils travel to sites of infection. Specifically, inflamed cells release chemicals that summon neutrophils. Once they arrive, the neutrophils anchor themselves to the inside of the blood capillary. They then dissolve a portion of the basement membrane, which allows them to squeeze out of the vessel, a process called diapedesis, and enter the inflamed tissue.
I think that is so cool how that actually happens, and such a great animation to actually show you the visual of what's happening there. So another part of non-specific immunity is antimicrobial proteins. We have two of those, interferons, and we have a complement system. These are really two different types of proteins which help provide non-specific resistance against bacterial and viral organisms. So let's learn a little bit more about these. So interferons happen or take place or play their role when a virus infects a cell. The cell produces interferon, which it releases to nearby cells. The interferon then binds to surface receptors on neighboring cells, and this triggers the production of enzymes within the cells that would prevent the virus from replicating if it managed to invade. So this cell kind of gets invaded, and it's now telling its neighbor cells, hey, watch out, be careful, we have this virus, and it sends them kind of this interferon, which is a protective mechanism. So if the virus comes in contact with those cells, it cannot become infected with the virus. The other piece of this is the complement system. So we have more than 20 different proteins called complement that circulate in the bloodstream in an inactive form. If a bacteria or antibodies against a bacteria activate the complement, a complement reaction continues as a cascade of chemical reactions with one complement activating the next. So we have this, kind of cascading event that happens here. So let's watch this animation. We can kind of watch this play out a little bit.
More than 20 different proteins called complement circulate in the bloodstream in an inactive form. A bacteria, or antibodies against the bacteria, activate the complement. This triggers a cascade reaction in which one complement protein activates the next. The final 5 proteins are called the membrane attack complex. These proteins embed themselves into the bacterium's plasma membrane in ring-like circles. This action punches holes in the bacterium. Fluid and sodium rush into the bacterium through the openings, causing the bacterium to swell and burst.
So again, just another kind of neat way that our body can kill bacteria. I think it's so cool. It really intrigues me that we have these built-in systems that kind of do this. Another part of nonspecific immunity is natural killer cells. So these are really, a unique group of lymphocytes that can recognize and destroy any foreign cells, including cancer cells, virus-infected cells, bacteria, as well as cells of transplanted organs and tissues. So sometimes we don't want them to be doing their job. They use several different methods to destroy the cells. They really roam throughout the body. Most of them involve the secretion of chemicals that cause the cell to die or break apart, which is called lysis, L-Y-S-I-S, lysis. So it causes the cells to break open, which then kills them. Inflammation is another piece of nonspecific immunity. So it's a pretty involved process. There's a lot of different pieces of immunity. A lot of different pieces of inflammation. Okay, so we're going to talk through all of these. But basically, inflammation occurs because of some sort of tissue injury. So something like trauma or cell death or infection causes the inflammation process to start. So what really happens is we get these injured cells secreting chemicals such as histamine that dilate blood vessels in the area. Blood rushes in, bringing leukocytes. Okay, the same chemicals trigger vasodilation and cause cells in the capillary wall to separate. Fluid, leukocytes, plasma proteins, antibodies, clotting factors, and complement leak out. Fibrinogen forms a sticky clot to keep the infection from spreading. We also get neutrophils drawn to the area, and so they can phagocyze pathogens. They also secrete chemicals to summon other neutrophils. Okay, so lots of different things here happening in the inflammation process. So let's watch the animation on this and get a little bit better of a visual on it, and we can talk a little bit more about what your book says about it.
Tissue injury, whether from trauma or infection, produces inflammation. Inflammation stimulates the body's defense system to begin fighting the infection while instigating measures to contain the pathogen. The inflammatory response also includes processes that clean up and repair damaged tissue. Injured cells secrete chemicals such as histamine that dilate blood vessels in the area. Blood rushes in, bringing necessary leukocytes. Increased blood flow also helps flush out toxins and wastes. The same chemicals that trigger vasodilation also cause the cells in the capillary walls to separate slightly. Fluid, leukocytes, and plasma proteins, as well as antibodies, clotting factors, and complement, leak through the spaces and into the injured tissue. Fibrinogen that leaks into the area forms a sticky clot that walls off the infected area, helping to keep the infection from spreading. Neutrophils, which have been drawn to the area by chemicals released from damaged cells, actively phagocytize pathogens. Neutrophils also secrete chemicals called cytokines to summon other neutrophils and macrophages to the area. Macrophages destroy bacteria. They also help clean up the area by engulfing damaged cells and dead neutrophils.
All right, so lots of things happening here. Both the immune system, the lymphatic system, and also our blood clotting system is coming into play here, right? When we have injury like this. Another thing I want you to know about the inflammation is there's 4 main signs of inflammation: swelling, redness, heat, and pain. Okay, so swelling results from, we get all this fluid there, right? We've all had some sort of injury to our finger or hand, whatever, part of our body. Redness and heat come from the increased blood flow, and then pain we get either from damage to nerves or the pressure, right? One time I shut my finger in the car door, and it hurt so bad the next day because there was so much pressure in there because all that fluid went to my finger. My finger didn't have enough room for it, and it was just causing all of this pain and discomfort. So lots of different things. We definitely need to know those 4 signs. Fever is another part of nonspecific immunity, also known as pyrexia. Pyrexia, or if somebody has a fever, they're said to be febrile. Okay, so fever is really a beneficial part of our immune system. It promotes the activation of interferon and inhibits the reproduction of bacteria and viruses. So we want it to happen. So there's really a sequence of events in a fever. So the first step is neutrophils and macrophages. They eat or engulf the bacteria, and then they secrete a substance called pyrogen. The pyrogen stimulates the anterior hypothalamus to secrete PGE, okay? Prostaglandin E. Prostaglandin E resets the body's set point for temperature. For example, it may raise it from a normal of 98.6 up to 102. When the set point raises, the body needs to generate heat to bring it up to that set point, and it does this through shivering and constricting blood vessels in the skin. This results in chills and cold, clammy skin, which we've all felt when we had a fever. The temperature rises until it reaches its new set point. It stays there as long as the pathogen is present. So when our body has taken care of the pathogen, or it's no longer a threat, the phagocytes stop producing the pyrogen and the body's set point for temperature returns to normal. So again, this is another negative feedback system, okay? And when the temperature returns to normal, the body needs to lose the excess heat, which it does through sweating. So that's why when people break their fever, okay, we see them break out into a sweat. So it's actually a good sign. Even though fevers are beneficial, excessively high fevers can be dangerous. So we do want to make sure that somebody's fever doesn't go too high, and especially in children, it's much more apt to cause things like seizures or coma for a high fever in children. All right, so lots of parts of nonspecific immunity. Let's talk about a couple of parts of specific immunity. These are a little bit more in depth, but there's less pieces to it. Okay, so we have cellular or cell-mediated immunity, and then we have humoral or antibody-mediated immunity. So these specific immunity in general is directed against a specific pathogen. So it's the opposite of nonspecific immunity. Nonspecific immunity is very general in nature, so we just get any sort of pathogen entering our body, nonspecific immunity takes over. Specific immunity only takes over when we've already been exposed to this particular pathogen before, and it uses these two different ways to destroy it. So cellular immunity just aims to destroy foreign cells or host cells that have become infected. Humoral or antibody immunity focuses on pathogens outside the host cells, and it kind of marks them for later destruction. Both processes use lymphocytes and antibodies. So let's first talk a little bit in more detail about those. Lymphocytes fall into one of three classes. Natural killer cells, we already talked about, T lymphocytes and B lymphocytes. T lymphocytes or T cells develop from stem cells in the red bone marrow. Before they're fully mature, they leave the bone marrow, go to the thymus where they continue to mature. We already talked about that. Once these T cells are mature, they leave the thymus and migrate to lymphatic organs and tissues throughout the body to do their job. B lymphocytes, on the other hand, stay in the bone marrow until they're mature. Okay, so one way to remember this, T cells mature in the thymus, which starts with a T. B cells mature in the bone marrow. Antibodies are the other piece of this. Okay, antibodies are also known as Ig or immunoglobulins. They're proteins formed by B cells. They're found in plasma and different body secretions. Okay, antibodies consist of a chain of protein in either a Y or a T format. Okay, the end of each arm of the Y is uniquely shaped, allowing each antibody to combine with a specific antigen. So you see that happening here. It's got a specific shape here. Only this antigen can fit into it. An antigen is any molecule that triggers an immune response. So any foreign substance is said to be antigenic. Okay, this is the system and how the body learns to identify itself from foreign substances. So this is our ability to kind of recognize foreign substances. There is different types of immunity. Okay, we have active and passive immunity. Active immunity is permanent, or at least it lasts for a long time. It's when the body makes its own antibodies or T cells. Passive immunity results from an injection of antibodies from another person or animal. Okay, it only lasts usually for a couple of months. Okay, so let's talk about these different classes. We have natural and artificial, active and passive immunity. So natural active immunity occurs when the body produces antibodies or T cells after being exposed to a particular antigen. So if you've gotten the flu type A, let's say, your body produces antibodies against this virus. And if you become exposed to flu type A again, your body will fight it off before you get sick. Artificial active immunity results when the body makes T cells and antibodies against a disease as a result of vaccination. So this is where vaccines come into play. So we get injected with a vaccine containing dead or weakened pathogens. Our body, though, still recognizes that, builds up antibodies against it. So if we do then come into contact with, let's say, flu type B, which we're immunized against, we will not hopefully get sick because our body has already built up antibodies. So when we're exposed, the antibodies fight off the infection before we actually get sick. Natural passive immunity results when a fetus acquires antibodies from the mother through the placenta or through breastfeeding. Again, passive immunity doesn't last for a long time, but it does help. Okay, so that's why breastfeeding, they always talk about breastfeeding. It's really good for babies because it helps to build their immune system because they get antibodies from their mother. Artificial passive immunity involves obtaining serum from a person or animal that has produced antibodies against a certain pathogen. So we're not like we're in vaccinations where we're actually giving somebody the virus so the body can develop their own antibodies. We're just giving them the antibodies, but they don't get a ton of antibodies. So that's why it doesn't last for a long time, and it's called passive immunity. Sometimes this is used for treatment of rabies or botulism. Kind of gets our immune system working a little bit faster so we can start kind of fighting off the infection while our body is in the process of making its own antibodies. All right, so let's get back to non-specific immunity, or specific immunity, and talk about the last couple of things, our cellular and humoral immunity. So cellular immunity really works to destroy pathogens within a cell. So 3 classes of T cells. Cytotoxic T cells, they carry out the attack. Also called killer T cells, but not to be confused with natural killer cells. Helper T cells play a supportive role. Memory T cells remember the pathogen in case future encounter. So let's watch the animation on this. Walk us through this step by step.
Cellular immunity destroys pathogens that exist within a cell. The immune process begins when a phagocyte, such as a macrophage, reticular cell, or B cell, ingests an antigen. The phagocyte displays fragments of the antigen on its surface. This process, called antigen presentation, alerts the immune system to the presence of a foreign antigen. The phagocyte displaying the fragments is called an antigen-presenting cell, or APC. When a T cell spots the foreign antigen, it binds to it. This activates or sensitizes the T cell. The T cell begins dividing repeatedly to form clones of identical T cells already sensitized to the antigen. Some of these T cells become effector cells, such as cytotoxic T cells and helper T cells, that will carry out the attack. Others become memory T cells. The cytotoxic T cell binds to the surface of the antigen and delivers a toxic dose of chemicals that will kill it. Helper T cells support the attack by secreting the chemical interleukins. Interleukins attract neutrophils, natural killer cells, and macrophages. They also stimulate the production of T and B cells. After the attack, some cytotoxic T cells and helper T cells become memory T cells. These numerous, long-lived cells retain a memory of this particular pathogen. If re-exposure to the same antigen occurs, these cells will be able to launch a quick attack.
So that sums up cellular immunity, a few steps there, but it kind of makes sense, right? And I hope you're starting to see that all of these processes are working together to some degree to fight off infectious processes. So the other part here is humoral immunity. So this focuses on pathogens outside the cell, okay? Uses antibodies to mark the antigen for later destruction. Doesn't destroy it directly. It kind of saves it It just marks it and tells the body then that we need to destroy it at a later date. So let's watch that animation here and see how this system works.
Humoral immunity uses B cells that contain thousands of receptors for a specific antigen. When an antigen specific to that receptor comes along, it binds to the B cell. The B cell engulfs the antigen, digests it, and displays some of the antigen's fragments on its surface. A helper T cell binds to the presented antigen and secretes interleukins, which activate the B cell. The B cell begins to reproduce rapidly, creating a family of identical B cells that are programmed against the same antigen. Some of these cloned B cells become effector B cells or memory B cells. Most, though, become plasma cells. The plasma cells secrete large numbers of antibodies. Antibodies stop the antigens through a number of different means, including binding to the antigen's attachment points to prevent it from attaching to a human cell, or setting off the complement cascade, which destroys the invading microorganism.
So lots of ways that these antibodies destroy it, but this is how antibodies are produced. So this is a really important part of our immune system and developing also immunity, right? We were just talking about vaccines and those types of things and developing antibodies. So our system has to be exposed to the antigen in order to develop the antibody. So remember, the antigen is like the pathogen, the virus, the bacteria, whatever we're trying to fight off. The antibody is then our body's way to protect ourselves in the future and also to help stop the present infection. The last little piece I want to talk about here is allergic reaction. So we're not going to talk about, a lot of parts at the end of the chapter where it talks about diseases, but we are going to talk briefly, just quickly, about allergic reaction. So upon first exposure, the body produces IgE, okay? Now the person is sensitized, okay? So usually the first allergic reaction isn't as bad. Upon subsequent exposures, the allergen binds to the antibodies, causing mast cells to release histamine, and that's when we actually get the allergic reaction. Sometimes it's, watery eyes, itchy eyes, swollen lips, and sometimes it's a full body anaphylactic reaction. And that can actually is how people die from allergic reactions. And often it's a really strong allergic reaction, but sometimes people think they've been exposed to something, so they're not allergic to it, but don't understand that this is actually kind of a multi-step process.
An allergy is a condition in which the immune system reacts to environmental substances called allergens. For example, ragweed pollen is an allergen. When someone with a genetic predisposition to an allergy, such as ragweed, is first exposed to the allergen, the body responds by producing large amounts of the antibody immunoglobulin E, or IgE, specific to ragweed. These antibodies bind to mast cells. Mast cells reside in the skin, the lungs and upper airways, and the lining of the stomach. Although this response does not produce an allergic reaction, the person is now sensitized to ragweed. When the person encounters ragweed at a later date, the allergen binds to the antibodies already in the body. If the allergen links two or more antibodies, the mast cells release histamine and other inflammatory chemicals. Histamine causes inflammatory responses that produce the symptoms of an allergy, such as runny nose, watery eyes, congestion, and hives.
And ragweed is something that most times people don't have an anaphylactic reaction to, but this can cause, certainly these same steps can lead to an anaphylactic reaction.